Okay, so let's take a look at problem 514. So in Drosophila, a cross was made between females, all expressing 3 X-linked recessive traits. Okay, so they are called, let's see, my pen's not working as usual. Let's see, okay, it should be working. Okay, Scute, Sable, and Vermilion. So we're going to abbreviate those SC, S, and V. Okay, so we're going to cross a female expressing all those traits. X-linked recessive traits they are, so the female must be homozygous recessive. We're going to cross all of them to a wild-type male, and males are hemizygous for all genes on the X chromosome. So I'm going to denote the male genotype like this. And we have a Y chromosome down here, and here is our male symbol. Okay, so that's going to be the P1 cross for this question. Now we're going to cross these, and the question tells us in the F1 generation all females are wild type and all males are all males express all 3 mutant traits. So they're SC, S, and V. And why is that? And you should probably already know why that is the case, but let me just map that out for you. I'm going to use the green color here. So the females in the P1 during meiosis can only make one genetically distinct type of gamete, one that has all 3 mutant alleles for these 3 genes. The males, however, can make 2 types of gametes. One carries the X chromosome with the wild type alleles. The other carries a Y chromosome. So if we were going to do a Punnett square, very simple Punnett square, what I would do is I would map it out like this. So only one column, and I would put the female gamete right here, and I would put one of the male gametes right here, and I put the other male gamete down here, and then we can fill in the square, the 2 squares I should say. So this right here is going to be the genotype of all of the females in the F1 generation, because they have 2 X chromosomes, right? And the females are all going to be wild type with respect to phenotype because they all have at least one dominant wild type allele. On the other hand, the males are all going to be mutants, right? Because they're hemizygous. They only have 1 allele for each of these genes on the X chromosome, and each allele for those genes is the recessive mutant allele. So the males are all going to be Scute, Sable, and Vermilion. So in the next part of the question, we see that the cross, so the F1 flies were crossed. So these females right here were crossed with these males. And let's see, maybe I can I'll map that out, diagram that for us right here. So we're crossing those F1 females with the F1 males and we're getting the F2 generation. In the F2 generation, let's see, do I have that? Let's see if I can, oh yes, here it is. And here's the F2 generation. So there are, I think, 1,000 flies here. To check that math in a moment. And the question gives us the phenotypes of those 1,000 progeny in the F2 generation. So 314 of them carry or have the mutant phenotype of scute, sable, and vermilion. 280 of them are wild type, and so on and so forth. I'll just choose this one right here. 156 of them have the scute phenotype, but are wild type for the other two genes. So what the question is now asking us in part A, it says, using proper nomenclature, determine the genotypes of the P1 and F1 parents. So we've already done that by going through the cross really slow, right? Or going through the question slowly. So here are the P1 parents right here. Here's the genotype of the female. Here's the genotype of the male. Okay, and then for the F1 parents, here's the genotype of the female. Here's the genotype of the male, and then I transfer those down here. So same thing, F1 parents, genotype of the female, genotype of the male. Okay, so part A is already done. Now part B is saying determine the distance, or no, determine the sequence or order of the three genes and map the distances between them. So essentially all that was setting us up to figure out what gene is in the middle using a 3-point cross. So in class today, this is September 27th. It's about an hour after class, and I'm trying to do these problems to get them up on YouTube for you all. We went over some basic steps of a 3-point cross, and what we talked about was okay. I can map these steps out for us here. Is we need to guess at the order of the genes. So in this case, let's go with SC, S, and V. Okay, we're going to go with that order. Step 2, we said now we need to do a cross, right? And we need to cross a heterozygous individual with a homozygous or hemizygous recessive individual, and that's what we've already done in this problem. And then we need to get the phenotype data of the progeny, and we've done that in this problem. So that has already been done for us. Excellent. And here's the phenotype data right up here. Okay, great. And there's the cross we did up here. Okay, so the next step we were supposed to do was determine the arrangement of genes, right? So the arrangement of alleles, that is, in the heterozygous female. And we've kind of already done that, right? Because we started with certain genotypes in the P1, right, in the female. So all the recessive alleles were linked together, and they moved as a single unit from the mom over to the sons in the F1 generation right here. And also over to the daughters right here in the F1 generation. So we kind of already have the arrangement. We already know it. And let's look at the parents. Right up here, this F1 female, we've already determined that all the wild type alleles are together and all the mutant alleles are together. So the arrangement of alleles is like this. And I'm assuming S is in the middle here. So this is the arrangement of alleles in the female parent. If you're confused on what arrangement means, well, you know, I can show you a different arrangement. So a different one, let's say, would have the recessive S allele with the wild type versions of Scute and Vermilion on one chromosome, and the wild type version of S or Sable with the mutant versions of Scute and Vermilion on the other chromosome. Now that's a different arrangement, but we don't need to worry too much about determining the arrangement for this problem because we've already figured that out. In class we went over a way to look at the progeny phenotypes to determine the non-crossover gametes, and that would tell us the arrangement, but we don't need to do that here. So now that we know the arrangement, we can predict the various classes of gametes, the 4 classes of gametes that should result from meiosis in this individual right here, in these female flies. So let me map that out for you down here. So I'm going to redraw the genotype. And draw the 4 possible classes of gamete genotypes that can result. So the first one is the non-crossover class. So I'm just going to diagram them like this. So this is one possible gamete that results from no crossover, and this is the other one. And you can see the reciprocals. Every time we see a wild type allele in this gamete, we see the mutant allele in the other gamete. So this is the non-crossover class. Now, here, what we're going to assume is that a crossover is happening between S and V. So the mutant allele is going up, the V plus is going down. So I'll use a different color here. Why not? So SC plus, S plus, little v came up. This is one gamete, and the other one is going to be the opposite. This is the other gamete. Okay, and this is single crossover. We'll call this class 1. Now let's take a look at the next one. So this one over here, the next class, we're going to assume a crossover is happening between SC and S. So the wild type allele is going to go down, and this one's going to go up. And ignore this over here. Okay. So SC, S plus, V plus is one gamete. SC plus, S, and V is the other gamete. And that's single crossover class 2. Okay, so now we have one more, the double crossover class. And let's use pink. Okay. So double crossovers. Now, double crossovers are going to result when there's a crossover on this side of S and a crossover on this side of S. So essentially, SC is moving up, SC plus is moving down, and V is moving up, and V plus is moving down. So let's just draw that. SC, S plus, V, and SC plus, S, V+. Okay, so if I didn't make a mistake, we have our 8 different genotypes, 2 in each class, each of 4 different classes of gamete genotypes. So in class, we talked about the next step was going to be to actually match up this data. So let's see. Okay, match. So I have to paste in the phenotypes of the F2 progeny from the question, and we can take a look at those. And what we see is that 314 of the progeny have a phenotype of a mutant phenotype for each of the 3 traits. So scute, sable, and vermilion. So because of the way we set up the cross, we know that all of those 314 progeny must have come from a gamete from the female with that genotype right there. So 280 of the offspring have a wild type phenotype. And again, because of the way we set up the cross, we know that all of those progeny must have been derived from a female gamete with the wild type, with a wild type allele for each gene. So 280. And let me point out this one right here. So 156 of the offspring have just the scute mutant phenotype. They're wild type for the others. So 100. Let's see if we can find those here. Those would be right here. So 156 of the progeny must have been derived from female gametes with this genotype right here. And then we can transfer all these numbers over. So 150 have mutant for sable and vermilion, so they must have come from here. 46 are mutant for scute and vermilion, so they must have come from here. 30 have the sable mutation only, so they came from there. 10 have the scute and sable mutations only, so they came from there. And 14 have the vermilion, so they came from there. Okay, so now we need to check our data. That's step number 6, and we can see that we have a problem. So our problem is that the smallest number of progeny appear in the single crossover class number 1. So there are more progeny in the double crossover class. And this is bad, right? So if our predictions were accurate, then we should see the smallest number of progeny down here in this class right here. And you have to remember that these predictions, the reason why these genotypes appear in the double crossover class down here is because of our original prediction that S, or the gene for the sable phenotype, was in the middle. So that prediction must be wrong. So now we need to go back, make a new prediction, either SC is in the middle or V is in the middle, and see if one of those can produce a double crossover, a pair of double crossover genotypes that are represented in the smallest amount in the actual data. Okay, let's give that a try. Okay, so I think the easiest thing to do is to kind of go all the way down here. Okay, so now I'm just going to make a new prediction. Okay, this prediction is that, let's see, SC is in the middle, okay? We'll go with SC. No, you know what? I'm going to go with vermilion in the middle. So now V is in the middle. Okay, now this is the F1 female. Now meiosis in the F1 female is going to produce 4 different classes of gametes. The non-crossover class, the single crossover class. So remember, S is going up, S plus is going down. This is one of the single crossover classes. So SC plus, V plus, S up here, SC, V, S plus here. Okay, now the other single crossover class, SC moves up, SC plus moves down, SC up, it's one gamete there, SC plus moves down. Okay, now we have the double crossover class where S is moving up, S plus is moving down, SC is moving up, SC plus is moving down. The other way you could look at that is just V is moving up and V plus is moving down. Either way is fine. So SC, V plus, S, and SC, V plus, S. I'll move that C closer to S here. SC plus, sorry, V, S plus. Okay, so we have non-crossover gametes. Single crossover 1. Single crossover 2. Double crossover. Okay, so these are our new predictions. And let's see if they are accurate. Can I just copy and paste? I can. Excellent. So here is our data. And what we see is, what color haven't I used? Pink. Okay, let's use pink. 314, again, should have come. 314 progeny came from that gamete. 280, when I say that gamete, I mean came from gametes with that genotype, obviously. 280 offspring didn't come from one egg. 314, okay, yes, okay. 280 came from gametes with that genotype. 150 came from a gamete with mutant sable and vermilion. Can you see that anywhere? 150 from there. 156 came from a gamete that had wild type alleles for vermilion and sable but not skewed. 46 came from a gamete that had mutant alleles for skewed and vermilion but not sable. 30 came from a gamete that had a mutant allele for sable but not skewed and vermilion. Did I do that? Okay, I messed that up, didn't I? Yes. 46 comes from down here. I hope you caught that. That meant you were paying attention. So 30 progeny right here came from gametes that had wild type alleles for skewed and vermilion. Okay, that makes sense. Now, that leaves, I'm just checking over to make sure I didn't make a mistake there. Sorry for the delay. Okay, let's take a look here. 10 progeny came from gametes that had mutant alleles for SC and S. and a wild type allele for V. Wild type allele for V. Okay, and 14 came from a gamete that was just mutant for V. Okay, so this is perfect. Our double crossover gametes here are represented in the smallest numbers in the actual data. So our analysis shows that V is in the middle. Okay, so this is the right order. So SC, I can put plus, I don't have to put plus, but I'll put pluses, V and S. So our map looks like this. We could also, we could put S, and that's the same as writing S, V, SC, because it doesn't matter. The only thing that matters from a three-point cross is what gene is in the middle. And we determined V is in the middle. So excellent work. Now we need to actually figure out the distances between S and V and SC. So to do that, trying to change back to the red pen. Okay, so for that we need to sum the total number of progeny that resulted from gametes that underwent a crossover between S and V. Okay, now let's take a look up here. Which classes, I should say, involved a crossover between S and V? I messed up with this. I reversed the order of the alleles here, and that's going to be confusing. So I'm just going to go back to put SC in front. So I'm going to cut this out of here. Oh, I didn't want that to happen. Okay. Let's see if it's getting worse. What is going on? I just want to color this. Okay, let's try that again. Sorry. Okay, so let me remap this. I wanted SC in the front. SC, V, and S. Okay, so let's start with this interval right here. So we need all of the classes. We need to know which classes produced contain gametes that were produced from a crossover between SC and V. And we can look up here and we can see that, well, this single crossover class resulted from a crossover between V and S. So we don't want this one, not yet at least. But this one over here, yes, this resulted from a crossover here. And so did the double crossovers. Remember, the double crossovers involve crossovers on both sides, so between SC and V and between V and S. So we need all of the gametes that arose from a crossover between SC and V. In this case, it's going to be 156 plus 150 plus 10 plus 14, and I believe I can do the math in my head here, 306, 310, 320, 330. Okay, so 330 divided by a total of 1,000 progeny, or 1,000 gametes in this case, or progeny, it doesn't matter how you look at it, equals 0.33 or 33% or 33 centimorgans. Now, to figure out the distance between V and S, we just need to go up here and look and find each of the classes that arose from a crossover between V and S, and we can see this class did right here, and the double crossovers did right here. So we have to add those up. 30 plus 46 is 76, plus 24 is 100. So we have 100 gametes that involve the crossover between V and S out of 1,000 total equals 0.10 equals 10% equals 10 centimorgans. Okay, so that's it. That's the answer to number 14. That was a long problem, but we covered a lot of aspects of crossing over, single crossovers, double crossovers, reciprocal classes, homozygous and heterozygous and hemizygous parents. So there's a lot of information right here in this problem. And now we've seen how to actually determine what gene is in the middle if you have 3 genes, and the distances between those genes. So I think that this is a fun problem. Okay, as always, come see me if you have any questions. Thanks.